Welcome everyone to this week's edition of the Extracellular Vesicle Club, and a very special edition it is. I'm very happy to welcome Andrew Hill, who is the past president, the immediate past president of the International Society for Extracellular Vesicles. He's coming to us from lockdown, as with all of our other presenters today in Melbourne, Australia. So we'd like to welcome our presenters. And before we get started, I'd like to just have a brief conversation here with Andy. And Andy, I'd like to ask you about how you first got involved in extracellular vesicle studies. What was your path from, that first moment of, I wonder what these things are, to becoming president of the International Society?
Wow, well, it's been quite a journey, and actually, the journey started with one of our presenters today, Laura Vella, who we were setting up her PhD project, and the project was based around looking at how prions, which are these infectious proteins that cause neurodegenerative diseases, travel from cell to cell. And we were thinking about how this could happen, and we were looking at things such as the GPI anchor on the protein, and then a paper came out from Grassa Raposo's lab showing that the prion protein exits cells on these things called exosomes. And we thought, this is amazing, this is really cool. So we started looking into this, and Laura had set up some really nice cell models of looking at how prions infect cells, and we started to do what everyone does, grow up large numbers of cells, find someone who has an ultracentrifuge and spinning down liters and liters of conditioned media. And because my lab also had an interest in other neurodegenerative diseases such as Alzheimer's and later on Parkinson's, as we started working on EVs and prions, we moved into AD and Parkinson's. So we were pretty much a protein misfolding lab at the time. And then as we started working on EVs, we got more involved in the cell biology. And then, of course, the landmark paper by Jan Lotvel's group on showing the RNA content in the EVs came out. We really sort of took that on board and started to look at whether we could diagnose these neurological diseases by looking at their RNA content. So it's been a real evolution, and we've done all sorts of things. By some of Laura's work was one of the first papers to show that EVs exist in CSF. That was a really interesting story. We were trying to do this from mouse CSF, and for those of you who know much about mice, you can't get much CSF out of a mouse. And Laura gave a talk at our institute, and we found that the veterinary school next door had an animal house that held sheep on the top floor, and they were working on sheep CSF. So all of a sudden we got enough CSF to start looking for EVs. So along the way, we've sort of worked on all different aspects of EVs. Early on, we were told we're looking at artifacts and we should try and find something else to work on, but we persisted, and it's been a fantastic journey and really great to see how the field has evolved. And I think it's one of these fields where you can learn about so many other biological systems just by working on EVs themselves. So it's a bit of a long answer to how we got involved.
No, that's great to hear about that voyage through the vesicles of the world. And it's interesting. You mentioned sheep. We used to have sheep on the roofs of some buildings at my institute as well, because we were working on viruses that have central nervous system manifestations, and so we had some sheep around too. I don't think we still do, but it's interesting to hear about those commonalities. But Andy, you were also one of the first people to get involved with the International Society for EVs and to take a leadership role. And you eventually then became the president of ISEV. So could you just comment a little bit about the evolution of that society and where it has led?
Sure. Well, I haven't shared the story with anyone, but how I got involved with ISEV was Jan Lotvel. This came from the Paris EV meeting, the very famous Paris meeting, which I wasn't able to attend. But a crowd sort of sourcing event started to happen where Jan and others were emailing out, sort of asking who's interested in setting up a society. And there were lots of discussions around what it should be called, and the term extracellular vesicles started to be bandied around. And then there was a call for people to nominate for positions on the board. And I was on holiday at the time in North Queensland, and it was raining for like the whole week. And I was binging Breaking Bad, which was annoying my whole family. And then I got told, you should find something else to do. And I said, oh, there's this email about nominating for a new society. Should I do that? And it was like, if that gets you off watching Breaking Bad for the next 3 days, go and apply for it. So that's what I did. And I thought nothing would come of it, but I ended up being appointed as a member of the first executive committee of ISEV. And it was just brilliant just to become involved in a group that had common interests in EVs, but also very disparate interests. And we were all working on very different things. And it was great to feel part of a community of something that was growing very rapidly. But also, I think we all recognized at the time, we were all facing various challenges in getting our work published. You know, that there were people who did think EVs were just junk and they didn't really do anything. So from the very beginning of setting up JEV, the Journal of Extracellular Vesicles, getting the annual meetings going, and providing that forum for researchers in the field to come together, present their work. And it's amazing, even though, you know, I work on neurological disorders, talking with people on cancer, we had the same sort of problems. So it was a really nice field to go to meetings where people were very open with sharing their research, because it wasn't so competitive, and really just sort of being part of something that was growing. And then from there, I became the executive chair of communications, and that was quite a lot of work, running the website, setting up the membership options on the website and things like that. And then the opportunity arose to where I put myself forward to be elected as president of ISEV, and that was a really amazing opportunity as well, getting to meet so many people, and again, really helping to sort of grow the society, which I think now we have over about 1,500 members. So it's grown dramatically over the last decade, and it's still a really fun area to be working in.
Well, thanks for your service, Andy. I think that we're all very happy that you took that time away from Breaking Bad and could share your expertise and your leadership with the community. It's really wonderful to hear that. And I guess now that you are you know, the past president of ISEV, I mean, I can't imagine that you're lacking for things to do, but you've taken up a new project. Can you tell us a bit about what you are doing next?
Sure. So yeah, I always like to have something to do, and it is still keeping me away from the TV. So my new project is working as the editor-in-chief of a new ISEV journal, which is called the Journal of Extracellular Biology. And it's really exciting because we're launching in 3 weeks from today. And really, this idea came about 2 or 3 years ago, and Ken Witwer and Mark Warburton were really key in bringing this idea together, where we were seeing submissions to JEV, that there were, you know, getting papers into JEV now is quite difficult. The standard of articles making it into JEV has really grown. And we noticed that when JEV started, we had a lot of those technical papers, methods papers, that aren't finding their way into JEV as easily now. So we thought there was scope for another journal to perhaps take on some of those papers, but also to take up a slightly broader theme of looking at other things extracellular, so extracellular particles, lipoproteins, things like that. So the ISEV board has worked for a while now on setting up this new journal called the Journal of Extracellular Biology. We were going to abbreviate it to JEB, but JEV and JEB sounds very similar. We're going to work out how we can sort of distinguish the two. But the plan is that this journal would be a good home for some of those methods-based papers, also reviews, products from some of the ISEV workshops, and also for papers that might not meet the priority of JEV could find a home in this new journal. So we're launching in 3 weeks' time, that the submission site will open then. And look, I invite anyone who's got any ideas of content for the journal. If you've got an idea for a review article, for instance, that covers a topic that there isn't currently a review out there on, please get in touch with me, and we can chat about whether it might be suitable for the journal. And so we also invite you all to submit your research work to JEB as well. And to kick off the journal, we do have some fee waivers for the first articles that will be published in there. So that's again another little bit of an incentive to publish in the journal. And we're hoping this is going to have as much success as GEV, which you've seen grow over the last 12 years. So there'll be some communications coming out very soon about the journal. And please get in touch with me if you have any questions or have some ideas for content.
Wow, what a great opportunity for people to get involved at the ground level, of I think what's going to be a very important new journal, and one that is going to help guide the field as Jeb did in the beginning of the International Society for EVs. So thank you, Professor Hill, for sharing your experience and then also your vision with us today. I'm going to now, I think we've just passed 100 participants, so we have over 100 people who are waiting to hear about extracellular vesicles in the brain. So I would just like to ask the audience to please put your comments and questions in the chat box as we go through the presentation. And then at the end of the presentation, we will allow unmuting and I will moderate a discussion. So Andy, would you please introduce our other guests today and get us started with the topic?
Sure. So thanks, Ken, and thanks for the opportunity to come and participate in this webinar today. And we've got three great speakers who are going to help present the paper that we're going to cover today. The first is Laura Vella, who is the head of the Extracellular Vesicle Laboratory. Laura's an American Alzheimer's Association Fellow, and she works at the Florey Institute of Neuroscience and Mental Health here in Melbourne. So Laura, welcome today. And I was wondering if you could tell us a little bit about your lab and what are the things you're working on at the moment?
Thanks, Andy. Thanks, Ken. So as Andy said, we're based in Melbourne, Australia. My lab at the Florey, we predominantly focus on neurological conditions. And we have, I suppose, 4 aspects to the work that we do. And the first is really discovery science, and that's examining the function of the EVs, particularly phenotype transfer between cells. We examine EV content and use multiple omic approaches to do that, and do that with fantastic collaborators such as Andy in regards to RNA and Leslie, Gavin, who you'll hear from today in regards to lipid and protein. The second aspect of what we do is we have a look at EVs from animal models, and we look at both acute and aged animal models and study phenotype transfer in vivo using those models. The third part of what we do is develop methods for EV isolation. So you'll be hearing from Kate today about the method we developed to isolate tissues from EVs. And I suppose the fourth piece of the pie, what we do at the Florey is I suppose working with clinicians and biopharmaceutical companies really to enable access to patient cohorts and really the translation of our research. So I'll hand over and let Gavin introduce himself, who has been really key to the work that Kate's going to present today.
Thanks, Laura. Morning, everyone, or whatever time is in your time zone. So I'm Gavin Reid. I'm a professor of bioanalytical chemistry at the University of Melbourne. I work just across the road, basically, from Laura and the Florey. My interests mostly are around the development of analytical methods, mass spectrometry-based methods for complex biomolecular characterization. We've been working in lipidomics for about a decade now, including in extracellular vesicles. I was fortunate to be able to present at what was a very surreal second ISEV meeting in Boston in 2013. It was a very strange week, where we were presenting some of our initial work on characterizing lipid compositions in colorectal cancer-derived, cell line-derived EVs. I've been working with Laura over the last couple of years and with Kate as a joint student between our labs to apply those same methodologies to get an understanding of the role of lipid metabolism in extracellular vesicles in neurological diseases.
Okay, thanks, Gavin and Laura. So perhaps, Laura, could you tell us a little bit about what the background is to the paper that we're going to discuss today?
Yeah, no problems, Andy. So as I'd mentioned, we developed a method to isolate EVs from tissue, specifically brain tissue, because we're interested in neurological conditions. With many of these conditions, particularly Alzheimer's disease, we know that lipids are they're dysregulated in Alzheimer's disease, and they're dysregulated early on in the disease. So we wanted to, I suppose, profile in depth the content of the EVs out of this brain tissue, with the idea that they can potentially then inform us of lipid-based biomarkers, but also help us to understand the function of these EVs in the brain. So Kate came on board approximately 4 years ago now, and has become really our expert in regards to developing and improving the methodology for isolating EVs out of tissue. And utilising Gavin's expertise has really developed her own, I suppose, skill set in being able to profile these EVs out of tissue. So today she's going to present the work that she's done on frontal cortex EVs in Alzheimer's disease.
Okay, thanks, Laura, and welcome, Kate. Good to see you. So Kate Su is a PhD student, as you've heard, jointly supervised by Laura and Gavin. So Kate, can you give us a little bit of an intro to your PhD project and tell us what excites you about EVs?
Yeah, no problem. Thanks, Andy. Thanks, Laura. Thanks, everyone, for having me today. So yeah, I'm Kate. I started my PhD last year, but I've been working in the EV field for about 4 years now. So my PhD project is about identification and characterization of extracellular vesicles in neurological conditions. So my project, one of my hypotheses in the project is to characterize the cargo's content in the EVs from various neurological degenerative diseases. And we just think that the cargo's content in the brain-derived EV will tell us the disease pathologies in the brain, and also by revealing the profiles of that EV, we could really just pinpoint the altered cellular signaling pathway in the EV and in the diseases. And we're also interested in to look at the functional roles of those BDEV in the brain in different neurological diseases. Yeah.
Okay,
so,
thanks, Kate. And what excites you about EVs?
Yeah, what excites me about EV, I think. Yeah, what excites me about EV is that they are like very small, they are on nanoscale, but they are so powerful and mysterious in the way that they carry so much materials in there, and that can trigger changes in cells or in tissues. And yeah, they're just very fascinating.
Okay, excellent. All right, so without further ado, we're going to ask Kate to give her presentation on the paper Characterization of Brain-Derived Extracellular Vesicle Lipids in Alzheimer's Disease. And it was a paper that was published in JEV earlier this year. So thanks, Kate, and over to you.
All right, thank you very much. So good morning, good afternoon, and good evening, everyone. Today, I'm very excited to share with you my work on the characterization of brain-derived extracellular vesicles, lipids, in Alzheimer's disease. And so towards the end of this talk, I will touch base on the current work, which is to utilize the mass spec lipid platform to inform and to develop methods for plasma small EV and for lipid biomarker studies. So I'm based in the Neuroscience Institute, where hundreds of our researchers carry researches on neurological conditions. And the focus of my study is on Alzheimer's disease. The AD is the most common form of dementia, accounting for 60 to 80% of dementia cases. And currently there are about 50 million people living with dementia, and this number is expected to be tripled by 2050. And in Australia, an estimation of 500,000 people are living with dementia, and dementia has become the top leading cause of death in Australia. So AD has three broad phases: the preclinical AD, the mild cognitive impairments due to AD, and dementia phase due to AD. So to diagnose AD, neuropsychological and pathophysiological assessments are usually carried out, and the diagnostic paradigms include memory and cognition examination, evaluation of the biomarkers in the blood, in CSF, as well as PET scan. And however, due to the high cost and also the lack of resources, and not many AD cases are diagnosed, or most of the cases are diagnosed post-mortem. And unfortunately, we still have no effective treatment for this disease. So the current AD diagnosis paradigm is formulated around two fundamental the AD hallmarks, which is the extracellular accumulation of the amyloid beta plaques, and also the intracellular accumulation of neurofibrillary tangles formed by the hyperphosphorylation of the tau species. So other than these two hallmarks, there are many others essential features in Alzheimer's disease, and which include neuroinflammation, brain atrophy, oxidative stress, molecular peroxidation, and many others, as well as the lipids dysregulation. So with the purpose to figure out the cellular signaling and all the mechanisms that implicate AD pathogenesis, and also to identify potential biomarkers in AD. So my focus of my AD study is on the lipid dysregulation. So what are lipids? Lipids are building blocks for cellular membranes. They are a group of molecules composed of fatty acids, glycerophospholipids, sphingolipids, lipids, glycerolipids, and others. And lipids are involved in the maintenance of membrane structure and function, energy storage, and intracellular signaling. And lipid metabolism is involved in AD neuropathogenesis. So early events in the AD, such as oxidative stress, also as well as the ferroptosis, they cause lipid metabolism dysregulation and generate lipid peroxidation products. And also dysregulation of lipid metabolism has been discovered in AD brain tissues and in CSF, as well as in the blood. However, to access the lipidome profile in the brain tissue is difficult. So the small extracellular vesicles, most of us called exosomes, came into our view. So we are all familiar with small EV, and they carry cargoes such as proteins, lipids, nucleic acid, and metabolites that can report on the cellular regions, and they also play a role in intercellular communication by inducing changes in recipient cells. There are many studies focusing on the EV proteomic and genomic contents, and also their biomarkers potentials in diseases. But the EV lipids catches less attentions, but in my opinion, small EVs are very rich of lipids, and they're highly regulated by lipids. So we were thinking that the small EV lipids, they are important, and so we and also small EVs that possess typical lipid signatures, such as phosphatidylserine, cholesterol, sphingomyelin, etc. Although their lipid profiles vary depending on their cellular regions. So here I put up some of, but not all of the important EV lipids studies, and thanks to people such as Alicia Laurent to Scotland and many other teams that are working on the EV lipids, and they really push the field from going forward. And now we are starting to have a clearer ideas of EV lipid content, and many are still investigating of the potential of using this EV lipids as disease biomarkers. So back to my study, inspired by all these previous studies and coupled with the fact that small EVs are found in virtually all tissues, and small EVs are predicted to participate in AD pathogenesis. So small EVs, they are prime source for non-invasive AD biomarkers identification. So we hypothesized that brain-derived EV, especially their lipid content, can reflect and report on and give us an insight on the lipid changes in brain. And in general, the BDEV lipids could inform on the potential functions of EVs in neurodegenerative diseases, and they could review the pathways that are altered in the neurodegenerative disease, as well as they can be used to aid in the development of a molecular diagnostics tool. So to examine our hypothesis, we carried out the following study to isolate brain-derived EV from human frontal cortex in AD and control subjects. We used this rigorous BDEV isolation workflow developed by Laura a few years ago. And this approach involves dissociating the tissue without causing excessive cell disruption, which is very unique and also the key to its success. And I will take you through this process now. So essentially, we took the frozen frontal cortex, we gently sliced it and treated and dissociated the tissue with collagenase type 3, which is commonly used to dissociate cells from human or animals tissues that are very sensitive to damage. And the dissociated tissues were then subjected to a 300G spin for 5 minutes and 2000G spin for 10 minutes, and as well as a 10,000G spin for 30 minutes. And then we collected the 10,000G supernatant and loaded on top of this sucrose density fractions, which is composed of 3 different sucrose concentrations, and that were made up in the heavy water. And this gradient was then subjected to ultracentrifugation at 200,000G for about 3 hours. And at the end of the spin, there are 3 different vesicle fractions, which we call F1, F2, and F3, and their densities are written on the side here. And we then collected these 3 density fractions, which have different vesicles in there, and we collected them individually and spun them individually as well to palette the vesicles. And here F2 is enriched of brain-derived extracellular vesicles, and then we took this F2 fraction as well as the starting tissue materials for the subsequent analysis. And so this study included 8 AD and frontal cortices and 8 of the age and sex matched control cases. The derived BDEV were characterized by Western blot and transmission electron microscopy, TEM. So here in the Western blot, we could see that the BDEV in F2, they are enriched in the 2 exosomal markers, TSG101 and the syntanine, while the negative marker connexins there was very minimum signal or barely any signals there, indicating that the success of isolating BDEV with minimal cellular contamination. And the TEM image here shows the typical cup-shaped and the morphology of BDEV, and also the size of these vesicles were between 50 to 150 nanometers in diameter. And after the BDEV characterization by Western blot and TEM, we submitted the samples to this comprehensive lipidome analysis workflow developed in my co-supervisor, Professor Gavin Reid's lab. Firstly, the samples, the brain tissues also, and as well as the BDEV was subjected to monophasic lipid extraction, in which we spike in a number of deuterated lipid internal standards during this extraction process to account for lipid extraction efficiency, and which then allows for accurate lipid quantitation. We also We also performed chemical derivatization on the aminophospholipid, such as the phosphatidyl-serine, as well as the phosphatidyl-ethanolamine, and also the plasmalogens lipids to resolve isomeric lipid mass overlap, as well as to enhance the ionization. And then lipids were delivered by this nanomate, automated nanomate with an electrospray ionization source and analyzed in this Orbitrap mass spectrometer using the shotgun method. The requisition time for the direct infusion method was 3 minutes for each samples at both positive and negative modes. And for certain lipids, we also performed tandem mass spec to obtain the fatty acid chain compositions in the lipids. And then all data were analyzed in Lipidstage and as well as the in-house built R script, and the statistics was done in GraphPad. So we recently published this extensive lipidome workflow as a book chapter and method in molecular biology. So in this chapter, we specified the amount of internal standards that are required for different biological samples, including brain tissues, the BDEV, and different cell lines, as well as E. coli. So for the audience, if you're interested in doing lipidomics, please feel free to look this up. So after stringent data filtering and analysis, we managed to identify a total of 692 lipids spanning 4 different lipid categories and 17 lipid classes for downstream analysis and comparisons. So from the lipidomic data, we first looked at the lipid compositions in the tissue and also the derived brain, the derived EV isolated from these tissues, and regardless of the disease conditions. So by looking at the lipid class abundance distribution here, we found that BDEV were, this in light blue here, they were enriched in phosphatidylserine, which we also call PS lipids. And this is aligned with most of the previously published cell-derived EV. And PS lipids on the EV membrane are proposed to be involved in the intercellular communication, and they play a role in modulating EV uptake by recipient cells. And moreover, we determined that BDEV have a distinct PS subclass profile, which is that they are enriched in ether PS lipids, which is the alkyl PS, the PSO here, and also the alkeno PS, which we also call the plasmalogen PS, the PSP here. So, and also understanding the ether PS subclasses associated with the EVs were previously limited due to the lack of the sensitive analytical platforms. And our comprehensive lipidomic platforms here, including the derivatization step, and as well as the highly sensitive mass spectrometry, we were able to identify the PS subclasses, the PSO and PSP, as well as to look into their some composition levels, and therefore increase the PS lipids identifications. So why do we care about ether lipids? Let's have a look at the structure of this lipids using the phospholipid as an examples. So unlike most of the phospholipid, the diisophospholipid, which have 2 ester bonds at SN1 and SN2 positions that can link to the fatty acid chains, the alkyl ether ether lipids here, and instead of having an ester bond, they have a ether bond here, while the plasmalogens, which is the alkeno lipids, they not only have the ether bond, but also have the double bond next to the ether. And the ether lipids could change the, because of the structure is different, they can change the membrane structure, the stability, and the permeability. And studies show that plasmalogen species, they can facilitate membrane fusions, which in the case of brain-derived EV, they may implicate increased small EV uptake or fusion with recipient cells. So other than modifying lipid membrane structure, because of the presence of this double bond in the plasmalogen lipids, the plasmalogen species are prone to oxidative stress, and the plasmalogens are abundant in the central nervous system, and they can accommodate polyunsaturated fatty acids, and they may serve as an antioxidant and then play an important active role in scavenging oxidative stress in the brain. And I'll show more relevant data in the upcoming slides. So after looking at the lipid content in BDEV as well as versus the tissues, we analyzed the tissue data and found out that with n equals 8 for each subject, there was no significant difference between the AD and control frontal cortices at the total lipid level, the lipid category levels, or the lipid class level when normalizing the lipid data to tissue weight or even to the total lipid content. And we looked at if AD has changed the lipid compositions in the BDEV. We found there was a remodeling in the phosphatidylethylamine, the PE lipids in AD BDEV, as seen here in the subclass level and also the subcomposition level. So literature has suggested multiple, so here we can see that there's a downregulation of the diacyl PE lipids in the AD BDEV in the bright blue color bar here, accompanied with an upregulation of the plasmalogen lipids in the AD-BDEV. And so literature has suggested that plasmalogen can act as radical scavenger, as I just mentioned earlier, and they could modify membrane fluidity, which have an impact on EV uptake. And also a recent paper proposed that the polyunsaturated fatty acids containing plasmalogen species, they can make the cells susceptible to ferroptosis. So another finding in the PE lipids is that AD-BDEV recapitulates the deficiency of DHA in the brain by showing the decreased DHA, this 22,6 containing PE species, which is the LPE 22,6 here, and the PE 46 here. And while we could detect some of these changes in AD tissue as well, in some instances, the differences were greatly enhanced in the BDEV compared to tissues. For example, here the navy bar shows the control tissue, and the gray bar here is the AD tissues. And there was no differences between AD versus control tissue in these LPE 22,6 species, but there was significant reductions when we look at the BDEV. And so there was also greater differences in the BDEV between the AD versus control compared to the AD versus control tissue as well. So this indicates that the utility of BDEV for enhancing lipid signals and enabling the detection of differentially expressed lipids in the disease. And we also observed some remodeling in the sphingolipid metabolism in AD-BDEV. So here we found that the long chain containing sphingomyelin and ceramide, which is the sphingomyelin 36-1, sphingomyelin 38-1, as well as the ceramide 36-1, were significantly downregulated in the AD-BDEV. And there was also but then there was an increase in the very long chain containing sphingomyelin, which is the 42-2 here, containing the 24-1 acyl chain. But then this was offset by the downregulation of the sulfatide 42-2 that contains the same 24-1 very long chain acyl chain. And this suggested that there might be a remodeling in this galactoceramides pathway involving the very long chain fatty acid species. And also the significant decrease in sulfatides, predominantly the most abundant sulfatide 42.2 here in the brain is observed to be downregulated in both the AD tissue and the AD BDEV. And sulfatide clearance has been reported to occur in preclinical AD brain and CSF as well. So conclusion, what do we know from this BDEV study? Firstly, we introduced a highly sensitive and comprehensive lipidomic mass spec analytical platform that allowed us to review the comprehensive profile of PS lipids in the BDEV, and which then informs the structure that may affect EV functionality. And this platform can be used to inform on the PS profiles of EV from other sources as well. And we show that BDEV are enriched of PS lipids, and more specifically the ether PS lipids, the PSO and PSP. And we discussed about the functions and the roles of these ether lipids. We found that the lipid composition of BDEV could differentiate the AD from control, and there are enhanced plasmalogen P species in the AD BDEV, and there are downregulation of the BHA containing P species in the AD BDEV, and also there might be remodeling in the sphingolipid metabolisms here. And so this, we also discussed the potential function and also the potential of these lipids as the biomarkers potentials for AD. So altogether, this study illustrates that EVs enables improved detection of disease-relevant changes compared to the brain tissue, which is the NEAT samples. And this results establish a foundation for the investigation of BDEV lipids as potential biomarkers for AD diagnosis and also for their functional roles. So for the time remaining, I want to touch on my current work and talk about utilizing mass spec lipid platform to inform and develop an improved method for the isolation of EVs from plasma, and also to determine and evaluate the purity of plasma small EV. While others have reported on lipid profiles of plasma small EV or sera EV, and they haven't successfully depleted lipoproteins or performed in-depth lipid profiling. So therefore, the true lipid contents of EVs in plasma or sera remains unclear. So the isolation of pure small EV from blood remain a challenge, and mainly because there is a presence of massive lipoproteins. So lipoproteins and EV, they share common features, as you can see from this figure, adapted from this paper. And EV have a very similar density to high density lipoproteins, and they have a similar size to the very low density lipoproteins as shown here. And based on the conventional EV isolation methods, they're always co-isolated, and lipoproteins are enriched in lipids such as triglyceride and cholesterol ester, and which when co-isolated, they will significantly interfere or compromise the true lipid profile of the plasma small EV. So in order to isolate and purify EVs from lipoproteins, I adopted the method from Karen, Mr. Young-Leveld and Cecilia's paper publications a couple of years ago. And these methods combine the density fractionation and the size exclusion chromatography. So basically, the platelet depleted plasma was loaded on top of the OptiPrep density and spun for about 2 hours. And after the spin, the majority of lipoproteins are separated from the high density band, which contains the small EV and the high density lipoproteins. The high density band here was then collected and loaded on top of the this Cephorus crosslink 6V size exclusion chromatography column, and which allows the separation of the high density lipoproteins from small EV. And in order to characterize the lipid composition of the LDL, I also put this low density band onto this SEC columns and collected the LDL particles. And in the original paper, they have carried out the Western blot and proteomics and analysis to confirm the enrichment of small EV and also and to confirm the depletion of the lipoproteins. So here, here I'm showing you some preliminary data, lipidomics results from of the plasma and also the small EV, as well as the low density lipoprotein that I collected from this the above workflow. So the previous mass spec lipidomic analytical platform was applied here again, and to characterize the lipid content in these three fractions. And in this preliminary data, we can see that there was an enrichment of the single lipids, mainly the ceramide here in the EV compared to the plasma or the low density lipoproteins. And EV also contained much less of triglyceride or the cholesterol ester compared to the lipoproteins or the plasma. And our comprehensive, highly sensitive and comprehensive analytical lipid profiling platform highlights the importance of lipoprotein removal for enhancing plasma small EV signals. And this platform can be used to validate or determine the current existing protocol of the efficacy of SEV isolation and depletion of lipoproteins. And therefore, it can be used to review the true lipid profile of plasma small EV, as well as to aid in the optimization or the development of more rigorous high throughput plasma small EV isolation techniques for the purpose of identifying small EV lipid biomarkers. So towards the end, I would like to thank everyone on this page who are involved in the AD-BDEV studies, and I would like to thank my supervisor, Laura Vella, Gavin Reid, Kevin Barnums, who really provided guidance in the past 4 years for this achievement. And thank you very much for listening. I am excited to hear your feedback and have a discussion with you all.
Thank you so much, Kate. That was a phenomenal presentation, and we're really grateful to you for giving it, and to all of the co-authors who contributed to this wonderful work. I just want to start by asking you about what you just presented, and that was on the plasma lipid profiles. How much material did you need for that? I might have missed that.
So with the method that developed in Cecilia and Jan Leber's that papers from a couple of years ago, we started with 6 ml plasma, and at the end of that process, the opti-prepped fractionation and also the size exclusion chromatography, and we had about 200 to 300 microgram proteins presenting in the EV fractions, and we took roughly 100 micrograms for the lipidomic analysis.
Mm-hmm. Great. Okay. So, and if you were to just target a few of those lipids, would you be able to start with much smaller volumes or not?
I think we for the lipidomics, the platform that I just showed you, the minimum requirement, I would say, it's 50 microgram of proteins. Yeah.
So we started with that large volume, Ken, simply so that we can, I suppose, create a reference. So an EV plasma lipid reference, so that I suppose the EV community will know exactly what the lipid profile of EVs is. So yes, while we started with large amounts, Kate can now reduce that significantly and is working on the development of various kits and methodologies so that we can create a system whereby we're using 500 microliters to a mil of plasma, it's a lot more high throughput.
That's great, because I mean, being able to understand where our signal is coming from is extremely important. So this is really interesting to hear. So let me go to the chat box now. And at this time, I would ask anybody who's given a question here to please get ready to unmute yourself when I call on you, so that you can talk with our presenters here today. So let's start out with Miguel Flores Belver. Miguel, I think you were off for a little bit, but you might have come back on. So Miguel, are you here? And if so, please unmute yourself and go ahead.
Yes, Ken, thank you.
Oh, great.
Thank you so much for the presentation. It's awesome work that you're doing, and it's really helpful for everyone. Thank you so much. So the question that I have is, I think looking at the first protocol that you published in 2017 and your presentation today, I was wondering if when you store the brain and then thaw it, if you are also, if the cells are damaged, the question is, are you able to isolate the EVs and then also pellet the cells in a healthy way, like intact cells? And if you don't, how could this affect to your EV population?
I can touch on that if you like. There are a couple of questions in the chat box regarding the methodology. So probably first up to note, if you're isolating EVs from tissue, it's going to be very difficult to get pure EVs. And I think that's why we've always used the term enrichment. So by using these methodologies, we enrich for tissue, enrich for EVs. Unlike working with conditioned media or fluids, there's a lot of variables to consider when you're working with tissue. Yes, there are going to be cells that are lysed. Yes, we try and minimize, I suppose, contamination as much as possible. And there are ways in which you can do that. So with CAN, you can modify the original method that we published by adding on a size exclusion step, and that does help a little bit. There was a suggestion in the chats as to basically doing an immunocapture. So that's another way. So you could do the size exclusion or the OptiPrep gradients, and then do an immunocapture after that. There's a couple of things to note. For example, the amount of collagenase you use, the batch of collagenase you use. We've spent so long testing out different batches of enzymes to determine which is most suitable for which tissue. And you wouldn't think it would make much of a difference, but boy, it does. So if the companies you're buying your enzymes from offer you the option to test different batches, try it out. Because we spent a long time not being able to isolate EVs, effectively, because the collagenase is just digesting away, a lot of digesting away, basically breaking open the cells and digesting away a lot of the proteins and associated with the EVs. Now, did that touch on your question? And were there some other questions in relation to the protocol?
Yeah, thank you.
Yeah, so I think we had a question. So Michael Klingeborn was asking about the possibility of doing maybe an immunocapture. Is that a method that you've explored?
We don't. So we use immunocapture for some other methodologies, but I can't see it should work here. I can't see why it wouldn't. So I would suggest I wouldn't do the immunocapture on that 10,000 G supernatant. I'd first do take the 10,000 G supernatant, do your size exclusion or your OptiPrep gradient, take the vesicles that you subsequently get from those techniques, and use those vesicles or immunocapture those vesicles to try and really pull out or enrich as much as you can for your extracellular vesicles, and remove, microsomal contaminants, for example, which, depending on, again, the tissue that you're using, the source, is inevitable. You may get some microsomal contamination.
Yeah, I think Michael's question is about maybe more general capture, but I would just note that Laura, using the method that you taught us, you and Andy, Tanina Arab in my lab has been doing capture of microglial EVs. And so she's using, you know, a marker that's enriched on the microglia. And so it is certainly possible to do it, but we haven't tried to do it, you know, where you capture everything. So yeah, that's great to hear. So then there's a question here from Krishna Maramanda. Krishna, do you want to ask your question about the collection?
Yeah, sure. Thanks, Ken. First of all, thanks for the presentation. So I have a quick question about the tissues, because I've been working in the tissues lately, like from the lung postmortems, and we have some couple of papers will coming soon too. I was wondering, like, because since you are showing some of the correlations with the blood and I mean, as it markers with the to the brain. So is it like a time of collections and all this important? And I mean, like at what time it didn't just any lipids that I mean, any inputs on that?
Cutting out a little bit there. Kate, did you want to address that? So is that, just let me confirm, is that the time of postmortem from the time from the time of collection? You're asking how much of an influence that has?
With respect to the postmortem of the tissue, and also the.
Yeah, sorry, Krishna, we're getting some breakup here, but yeah, so the postmortem interval, I suppose, and then yeah, and then what that would imply for human samples.
So we've always, Kate can comment to this in regards to what we've shown in her particular study. So what Kate had tried to do was make sure that all the samples had a very similar post-mortem interval. And Ken's group here, and probably members of his team on the chat as well, have actually examined different post-mortem interval times. So Ken, whether you wanted to actually comment on that, or maybe a member of your team could comment on that.
Yeah, so let's see here. I think Yao, are you on? You want to just make a comment here about postmortem interval?
Yes, can everyone hear me here?
Great, yep.
Okay, yeah, we actually did some experiment on the cat tissue, and basically when we put the sample in the room temperature for longer time, we compare the 2-hour versus 6-hour and the 24 hours, and we saw the degradation regarding the RNA content, and also we saw more intracellular proteins content in the samples with longer storage time. So I think basically we will suggest to take the samples fresh frozen after we took the tissues out from the animal models. But for the human tissues, it will be difficult when we got the samples from the brain bank, but it will be better if we can keep the original record about like the PMI samples for each of the brain tissue and all the organ we got.
Maybe comment on the potential impact on the lipid profiles regarding post-mortem interval. So obviously we'd be concerned about activity, particularly phospholipase activities in lipid degradation as a function of post-mortem interval. I think the key there is, and I think it's important to point out, that initially, while we want to know what the overall lipid composition is, we're mostly interested at this stage of defining what are the lipid profile differences between adjacent samples, Alzheimer's versus control. So provided they've undergone the same isolation strategy, including the post-mortem interval, any degradation in one group should be consistent with the degradation in the other. So while we can't control for that, there might be high levels of lysophospholipids, for example, or some head group degradation. As long as that occurs uniformly in both groups, we still should be able to see the differences between them.
That's a great point, Gavin. Yeah, wonderful. Yeah. So in terms of those lipid entities, we have a question from Patrick James. Patrick, I'm glad that it's not the middle of the night for you for once. Do you want to ask your question here?
Sure, thanks, Ken. So I guess this is probably more for Gavin than for Laura or Kate. So have you started looking at the localization of the lipids, outward, inward facing, and just your thoughts on how the asymmetry could relate to biology and diagnostics of EVs?
Yeah, look, I think that's important, particularly for the phosphatidylserine serine lipids. We know that they're a marker for an exon 5 recognition. So knowing, when we see an enrichment of PS lipids in the EV population, it would be nice to know if they're on the outer leaflet or the inner leaflet of the membrane. And of course, the proportion of that, because that might dictate its recipient cell recognition. There are ways in which we can tackle that. So actually using the same derivatization chemistries that we use to enhance our lipidomic profiling methods, we can use those same chemistry on the intact EV populations to get a sense for what is mostly for the PS lipids, but would also be suitable for the PE lipids, what the ratio of outer versus inner representation is.
Good. We have a question. I'm going to skip over one question and come back to it because we're talking about the lipid profile here and a possible artifact. So, Davide Trotti, do you want to ask your question about the atrophied tissue?
Yes, hi, good evening to everyone. I'm David Trotti from Philly. So I have a general question that actually goes to the problem of identifying control when you look at this postmortem tissue. And then what I would like to ask to the team is whether you think these changes that you guys see in the EV lipidomic profiles reflects rather a loss of the fact that you have a loss of neurons and atrophy in these heterophyous tissues rather than a disease signature, because what you're analyzing at the end is a tissue from a person that actually most likely lack all the neurons or most of the neurons. So what you really see is an EV profiles of what's left. And so the control becomes paramount. The control tissue becomes paramount in this comparison.
So I can, I suppose, touch on that to begin with. So yes, there is a concern if you're working with Alzheimer's tissue, you're going to have atrophied tissue. So yes, we do need to take that into consideration. And when we were first looking at our lipid results, that was one thing that we were, I suppose, careful to take into consideration. What was, I suppose, one thing that reassured us was the fact that we were seeing decreases in DHA, for example, which is a well-known, is really well known in Alzheimer's disease, both in lipids and in brain tissue. Excuse me. And also the signatures that we were identifying weren't, I suppose, indicative of atrophied tissue, and that they'd been seen previously in relation to Alzheimer's disease. Gavin or Kate, did you want to follow up there at all?
Yeah, I have a point to add on, which is that when we were looking at this, the EV secretion from the tissue in AD and control, we actually found that the EV released or isolated from the AD tissues were about half. The population was, or the protein content was halved compared to the EV isolated from the control tissue. And we have no idea what could be the cause. And so when analyzing the lipid data, we didn't normalize the lipid contents against to the starting material, the tissues, or so instead of normalizing that the lipid content in the EV to the starting material, we actually just normalized the lipids against the total lipid contents. Yeah.
Can you identify the control? What do you mean? What is to you? What is control to you?
The control tissue is just the with no AD pathology, and they were just the age, sex, and they're all matched.
We've also developing the profiles from other degenerative conditions. So if atrophy was an issue, I suppose we'd see a similar profile with other degenerative conditions that are associated with neuronal loss.
Okay, well, folks, we are over our hour here, but we do have 2 more questions in the chat box that I'd like to get to if the presenters are willing to just stick around for another 5 minutes. So if that's the case, let's go to Mariart Imanbekova. Do you want to ask your question about A-beta? So now we're getting into some really disease-specific things.
Hi, thank you, Professor Witwer. Thank you, Kate. Thank you, T, for wonderful presentation and great work. I have a question regarding the A-beta association within the lipid membrane. Did you confirm the association of beta within the EVs that you isolated? And do you think the A-beta may, may interact with membrane and change the lipid structure? Has it, like, I recall that it has been shown that beta may affect the lipid structure in the literature, but not within EVs. And yeah, in our work, we also found the similar results. So I was just wondering about this in your work.
So I can comment on that. So the association of A-beta with EVs, look, it's very controversial. I suppose I can talk to what we have seen in our hands. In our hands, we do see A-beta associated with a vesicle population, but it is not this vesicle population that I'm talking about. So it's not that Kate has been talking about. So it's not the vesicle population that's enriched in, you know, syntinin and TSG1 and has a similar profile to exosomes. We find A-beta associated with another vesicle population. I think you are probably correct in that, you know, A-beta loves membranes, it loves lipids, whether it can, you know, remodel and affect those membranes, we haven't looked at that ourselves.
There is a body of literature going back probably 10 years or more on looking at the interplay between A-beta and particularly cholesterol and sphingolipid metabolism, and that looks like that is part of a feedback mechanism. So it might not be unexpected that alterations in A-beta concentrations cause change in lipid abundance and vice versa.
I see there's a question here from Dr. Klingeborn. Do you want to ask your follow-up, please?
Sure. Can you hear me all right?
Yes.
So Kate and maybe Andy or Laura can also weigh in. And I did see Ken added in the chat there about this recent Michael J. Fox Foundation workshop. So what do you think is the current best way to get to brain-derived EVs from systemic circulation, given the recent paper from David Walt saying the L1 cam is not a brain-derived EV marker?
Yeah, initially my PhD project was set out to isolate brain-derived EV from plasma, and since now the more that we read in the literature and the more that we looked in the research field, I don't think it is feasible now to isolate brain-derived EV from the plasma. Yeah, and I think Andrew and Andy's lab, they probably have Andy probably has more to say about the isolating of neuron-derived EV from plasma.
Yeah, I mean, we've been trying, and you know, part of starting this work on looking at the brain-derived EVs was to try and identify some new markers that we could use in the circulatory system to pull out neuronal populations. But we haven't been successful so far. And I think we just have to remember that, you know, in the circulatory system, EVs are going to be coming from so many different places. So the number of neuronal EVs is likely to be very small. So I think it's still a hot topic really around how we can isolate neuronal derived EVs, or in fact any kind of tissue derived EVs. And I think that's where the field is really exciting with these protocols for isolating EVs from particular tissues or cell types, because I think that hopefully if we could build an atlas of that, we might be able to get some very specific markers we could pull out from the circulatory system where EVs that come from specific tissues.
Yeah, and so we've looked at, I suppose, the proteins that are extracellular on these vesicles and come up with a few hits that would, I suppose, identify the vesicles as being brain specific and not expressed in other tissues. So yeah, we're looking at that, but I think as the technologies sort of develop in regards to looking at single EVs, and I think as the technologies develop, we may be able to investigate this a little bit further in a bit more detail.
Thank you.
I agree with what's being said here. You have to think about how many, like what Andy said about the numbers, the stoichiometry, and then what your platform is actually capable of. So, we use the ExoView platform, for example, this SPRS platform, where you're capturing things onto a surface with antibodies, and in each of those antibody spots, you can maybe capture a few thousand EVs. So if your EV is at a frequency that's less than 1 in 100 or so, that's not really going to help you to quantitate so well. And then you have to go on to multiple markers, trying to understand multiple multi variable signature of many vesicles. So it is, I agree, a very exciting area. And hopefully we will have some new capabilities, some stretching of our current capabilities as the next months and years unfold. And we had, I see there's one more question that came in the chat box, and that is the link between protein and lipid sorting. So of course, that's also quite interesting. We often think about protein sorting into EVs, but the lipids, how are the lipids influencing that protein sorting? So we heard about the link between A beta and the lipid metabolism. So there's certainly some clues, I guess, that we can get from this research that you've all done.
Yeah, so in terms of the link of protein and lipid sorting into EVs, actually, that is a part of my PhD project. So one of my PhD project is to review the protein and lipid profiles in BDEV in multiple neurological conditions. And yeah, we haven't done the proteomics on these EVs in the AD tissues, but we will be doing it later, hopefully soon, and together with many others neurological diseases as well. So what we do is a systemomics strategy, and so after that lipid extraction, we will take that the remaining protein pellets and use it for the proteomics.
Wonderful. So there's a lot more to come. And I think I can speak for all of our audience in thanking each of you, Kate, Laura, Gavin, and Andy, for joining us today. We wish you the best in continuing this research and expanding it. We'll look forward to what's to come, and thank everybody for joining today. We hope to see you on another EV Club coming up soon. So take care, everybody. Bye now.
Everyone, bye.
Bye.
Bye, everyone.